Hello friends, I am MD Harun al-Rashid and it's my YouTube channel Harun-biozone. Today in this video I will discuss about the vernalization process in plants for flowering. So what is vernalization? Vernalization means the vernalization term derived from the Latin word that is verna. Verna means spring, or vernus means of spring. It is a crucial biological process where the seeds or plant exposed to prolonged low temperature near about 0 to 10 degrees centigrade to induce or accelerate flowering. Okay, the vernalization is a flowering process, but it occurs in cold temperature when plants or seeds are induced in low temperature ranging from 0 to 10 degrees centigrade. And in this low temperature, the vegetative growth of the plant converted into reproductive growth or flowering process. Okay. Without vernalization, the plant shows delayed flowering. Okay. Delayed flowering. Without vernalization or low temperature treatment, plant shows delayed flowering. Suppose this plant is converted to vegetative growth to reproductive growth in 10 days. Normally. But in case of vernalization, when we treat with cold temperature, then 0 to 10 degrees centigrade, the flowering occur in near about 5 days. Okay. So crop development process is shortened by the vernalization process. This process was first discovered by Trofim Lysenko in 1928. First used this term vernalization, but according to Clifford, first observed 1857. He shows that winter wheat sown seed during winter, but flowering occur in summer. This is the normal process. Clear? If this wheat seed treat with low temperature 0 to 5 degrees centigrade for few days, the spring few days then sown in springtime, they flowering in summer. Okay. Normally, this is a normal process where the winter wheat sown seed during winter, the flowering occur in summer. Okay. But this seed do not flower during the springtime. But this is during springtime, if the seed is treated with 0 to 5 degrees centigrade, the vernalization process, then flowering occurs in summer. Okay, also spring seed show flowering. So by the process of vernalization, the winter wheat is converted into spring wheat. So it is called the spring wheat or spring wheat. It allows the plant to reach vegetative maturity before reproduction can occur. So what happens? By vernalization process, the vegetative maturity become rapid. Okay, after vegetative maturity, the plant converted to reproductive maturity. Condition necessary for vernalization. For vernalization, some conditions are required. First, age of the plant. In cereal plants like winter wheat, vernalization effectively only if the germination seed have received cold treatment, temperature treatment for sufficient time. In normal case, cereal crops, the winter wheat receive some cold temperature treatment for few days and flowering occur. But in case of biennial variety like henbane, the chilling treatment required for when the rosette condition plant remain rosette condition, okay, or 10 days of plant growth. When the plant age is 10 days, the vernalization process is effective. Appropriate low temperature. Normally, for vernalization require 1 to 6 degrees centigrade temperature. Okay, but it is mainly most effective during 0 to 4 degrees centigrade temperature. Below 0 degrees centigrade, this is non-effective. But more than 12 to 14 degrees centigrade, it is almost non-effective. Okay, so 0 to 5, maximum 0 to 5 degrees centigrade is most effective temperature for flowering, most effective. Below 0 degrees centigrade it is less effective, and below 5 degrees centigrade is less effective. Above 5 degrees centigrade this is less effective, but 12 to 14 degrees centigrade temperature it is totally ineffective. Oxygen is very essential for germination of seed. At least 0.2% oxygen is required for germination of a seed. Water or humid condition is essential for vernalization. Okay, without water, seed do not germinate or plant do not grow. So water is essential. Then light. We study in photoperiodism, the light is essential or crucial factor for flowering. So for vernalization, light is also required. Technique of vernalization. For vernalization, what do we do? Process. First, soak the seed. This is the bowl, and the seeds are soaked in water. Okay, soaked in water. Desired seeds is soaked in water. Allow it to germinate in process chamber. Okay, this subject chilling treatment. This subject is to do here the cold or chilling temperature is applied. Okay, then the moistened seeds are dried. Then after a few days, the moistened seeds are separated. Finally, the seeds sown in the field and germination occur. Winter wheat plant in autumn, flower in spring. Spring wheat planted in spring, but flowering in summer. By applying the cold treatment. Next, the mechanism of vernalization. There are two hypotheses present for vernalization. Okay, first hypothesis is the hypothesis of phasic development. This hypothesis is proposed by Lysenko, where three different phases are required for vernalization or flowering of plants. First, thermophase. Thermophase is a cold phase. The initial phase focused on vegetative growth. Okay, leaves and stem, preparation of for flowering, because without vegetative maturity, the flowering or reproductive growth does not occur in plant. So, if we need the flowering of plant, the first require the maturity or reproductive maturity of the vegetative growth. The thermophase or cold phase is required for completing the vegetative phase. Photophase, the later phase where the plant requires specific light condition, day and light. For long day plant, different light required. For short day plant, different light required. For day neutral plant, different light required. So first thermophase, cold phase is stimulate. The first cold phase. Cold phase, what is the function of cold phase? Vegetative maturity, and number 2, stimulate the flowering process. Then specific light condition is required for day length, especially during photophase. Day length is important because day length require for different plant. For short day plant, on the basis of day length, plant are divided into short day plant, long day plant, and day neutral plant. Then vernalization role. Cold treatment effectively completely accelerates the thermophase, removing the requirement for long winter, allowing the plant to quickly enter the photophase. Say for example, winter wheat, normally planted in autumn and experienced in winter, vernalization on thermophase, flowering in spring. In planted in spring, Mrs. Winter cold stay vegetative and would not flower until the next spring of her expression. Full winter again. What is the significance? The main significance of this hypothesis is, first, when we apply the cold treatment or thermophase, the vegetative maturity completed and stimulate different genes that are necessary for flowering. Okay. Then light phase for different type of plant flowering in different time. Number 2 is hypothesis in hormonal involvement. Clear? According to Parvis in 1961, formation of substance A, substance A is by some stimulus. Formation of substance A from its precursor, and when chilling treatment is required, it is converted into, that is thermophase. Okay, converted into B chilling treatment. Then B is converted into C, another substance C, and finally developed into D or florigen. T of vernalin. D means vernalin. Okay, and here vernalin is converted to F for fluorine. A vernalin then converted to florigen. And this florigen is transferred from the leaf to the apical meristem. Okay, and that convert vegetative to reproductive phase. Convert the meristem from vegetative growth to reproductive growth. Melcher also said in 1939, he proposed that chilling treatment induced the formation of new floral hormone called vernalin. When low temperature applied on plant, the plant produced the low treatment and hormone that is called vernalin. Okay, vernalin. This hormone is transferred to the vernalin produced in the leaf because the chilling treatment is perceived by leaf. Okay, when the vernalin is produced, it transfer to the other part of the plant, especially the vegetative bud. This hormone, he grafted a vernalized plant with unvernalized plant. He grafted. Suppose this is the plant. Okay, this is plant. This is plant, and this is another plant that is grafted. Okay, this is another plant grafted. This is non-vernalized plant, but this is vernalized plant or cold treatment plant, vernalized plant. Okay, when he grafted these two different plants, the flowering occur in non-vernalized plant. Because during cold treatment, the vernalin hormone is produced in this red plant. Okay, then it transfer to the when grafted completed, this hormone is transferred from red to blue plant, and this vernalin stimulate the production of florism or flowering plant where florism and flowering occur. Then epigenetic changes on gene expression or role of different gene for flowering by vernalization. What is epigenetic effect? Epigenetic means suppose this is plant leaf. Okay, suppose this is plant leaf. When we applied cold treatment, the leaf produce vernalin. Okay, vernalin that produce florism, and ultimately flowering occur. Okay. If we use this cold treatment at once and then remove this cold, okay, but this process, when the vernalin is activated, this process continuously produce. This is called epigenetic effect. The epigenetic means once we apply the stimulus, if the gene activate on time, then it is do not deactivate. Okay, if we stimulus removed. The changes in gene expression that are stable even after the signal that induce the changes is removed are known as epigenetic regulation. Okay, epigenetic regulation. One model for how vernalization affect flowering that are stable. Changes the pattern of gene expression in the mesh after cold treatment. Suppose during the high temperature condition, this FLC2 flowering locus C, this is a transcription factor that inhibit the flowering gene, that is FT1. FT means flowering locus T. This hormone stimulates the flowering process, but during the high temperature condition, the FLC that inhibit the flowering condition genes, this gene inactivate the FT gene, so do not flower occur, or the plant show the vegetative growth. If applied the cold treatment, the FLC gene or FLC1 1 and 2, the flowering locus C, the mad box transcription factor that inhibit, so activate the flowering locus T, flowering locus T2, this gene. Okay, these two genes are very important for flowering. Okay, and it stimulate the flowering process. Vernalization also activate the agamous-like gene, agamous-like 20, agamous-like 20 gene. Okay, this gene is required for the identify the meristematic tissue, identify the meristematic tissue. Clear? Then when activate the agamous-like 20 gene, the gibberellin hormone, the flowering hormone, gibberellin, and vernalin hormone also stimulate this agamous-like 20 gene. When vernalization occur, they activate the agamous-like 20 gene. Also, gibberellin and vernalin hormone stimulate this active gene. Okay. Then plant produce florism hormone. This florism hormone transport transfer to different part of the plant and produce flowering. So there are two genes, that is FLC2, that flowering inhibitor gene that activates during high temperature, but during low temperature the FLC gene is inactivated. Okay, when FLC is inactivated, the FT1 gene, or that is FT1 means flowering locus T, so flowering locus T gene activated, and it stimulates the flowering process. Another gene like agamous like 20 gene that activate, and it identifies the different meristematic tissue of vegetative bud. So, and in the presence of gibberellin and vernalin, this hormone stimulates the formation of the florigen. Florigen activates the flowering. Practical application of vernalization. Vernalization shortens the vegetative period and induces the plant to flowering. The cold treatment stimulate the maturity of the vegetative growth. Okay, accelerate the maturity of vegetative growth. So after vegetative growth, the flower shows the reproductive growth of flowering. It increases the cold resistance plant. Vernalization process stimulate the cold resistance plant, so we can cultivate in colder region, cultivate plants in colder region. It increases the resistance to plant fungal disease. During low temperature, fungus do not survive. So cold temperature stimulate or produce the disease resistance plants. Plant breeding can be accelerated. It also helps in crop improvement. In biennial plant, vernalization induces early flowering and early fruit setting, fruit formation and fruit flowering. Allow 2-year crop, biennial crop converted into 1-year crop or annual crop. Enable temperature plants to flower in warmer climates by artificial chilling of seeds. Winter plant is cultivated in the warmer climates by applying of vernalization process. So devernalization process, when the vernalized plant is treated with high temperature, the FLC gene, flowering gene, FLC gene activated and inhibit the FT gene, flowering locus T gene. Okay, so do not flowering occur, so no flowering occur. Temperature is the important factor for flowering of different plants. Thank you for watching this video. This is the end of the vernalization process. I think you understand the complete process of vernalization. Thank you.